After talking about nucleolus, I thought of continuing this and talk about vitellogenesis. You know that vitellogenesis occurs in primary oocytes of females. Primary oocyte is a diploid cell. It has to undergo cell division, that's reductional cell division, meiotic cell division. And cell division is, of course, a non-stop process. When a cell has decided to undergo division, it will not stop at any point. The meiotic prophase is a bit elaborate, and we divide it for our convenience into leptothene, zygothene, pachytene, diplothene, and diakinesis. Because a lot of things happen in prophase, so we earmark the different happenings likewise. The cell division, that is meiotic cell division, is definitely quite an elaborate process. When the primary oocyte reaches pachytene stage, it suddenly awakens to a new realization that, well, I'm going to divide, and I know that I'm not supposed to stop at any point, but what will be my ultimate fate? I'm going to become a haploid cell, and haploid cell means that ultimately I'll be transformed into an ovum, and the duty of ovum is to participate in fertilization, and once the fertilization has occurred, the early embryonic development will begin, and that process will also be a non-stop process. And development is, of course, dependent upon a number of things. First of all, energy, food, etc. These are two things of great concern. So, dividing that way would make no sense. I have to first of all store a considerable amount of food, because I'm going to be exposed to a new environment where there will be no food available for me. So I'll have to keep certain amount of food within me in my cytoplasm. And of course, I will have to do something so as to be able to store energy. Keeping these two things in mind, it begins to do something. The first thing it does is that the nucleolus begins to proliferate. You know that nucleolus is the repository of ribosomal DNA, that is the DNA which contains sequences for ribosomal RNA. Then this DNA has to be elaborated, amplified, because the number of copies must be available so that the transcription of this DNA occurs. As a result, the ribosomal RNA will be formed, and the ribosomal RNA must be available for the biogenesis of ribosomes, and ribosomes must be formed in considerable amount and adequate numbers, so that there may be adequate amount of protein synthesis which is required for protein biogenesis and protein synthesis, and also for the formation of yolk and so on. So the platform must be adequately formed, ribosomes must be formed. So for ribosome biogenesis, the prerequisite is nucleolar DNA amplification, that is the availability of large number of genes that encode ribosomal RNA. After this, you find ribosome biogenesis. The number of ribosome increases from 10 to the power 6 to the 10 to the power 12. The entire cytoplasm is filled with ribosomes so that protein synthesis may take place. When these two things have occurred, then there's question of mitochondrial biogenesis, because mitochondria must also be available in adequate number. Mitochondrion is something like a prokaryotic cell. It is endowed with mitochondrial DNA, mitochondrial ribosomes, and a number of proteins that you know about. And these things have to be made for the duplication or biogenesis of mitochondria. So, and these things occur in the primary oocyte at Pachitin stage. When all these things have taken place, then the primary oocyte resorts to what is called vitellogenesis, which means the formation of yolk. And you know that yolk is a lipoprotein complex. The protein part is made in the ribosomes, and the non-protein, that is the lipoidal part, is manufactured in the agranular reticulum. These two things, these two molecules are complex to give rise to yolk platelet, yolk globules, or yolk granules, whatever we call according to the morphology. So yolk formation takes on and reaches a plateau. The whole cytoplasm is filled with the amount of yolk that is required for that particular individual. In some cases, huge amount of yolk is required. In some animals, moderate amount of yolk is required. It depends upon the condition of the animal set in future life. Accordingly, the eggs are called, microelesithal, megaelesithal, and so on, depending upon the amount of yolk they contain. So when yolk formation has stopped, that is completed, only then the nucleus begins to think of doing further cell division, which was, of course, arrested at Pachitin stage. It moves on. For further activity, there comes a clue from inside, from outside, and this leads to the dissolution of nuclear membrane. You know that at the end of prophase, the nuclear membrane dissolves. The nuclear membrane of primary oocyte, which is of course irregular in outline as a result of huge amount of activities carried on by nucleus. So it's the nuclear membrane becomes irregular in outline. The nucleus itself becomes voluminous. So the entire structure is called germinal vesicle. And when nuclear membrane dissolves, we say that germinal vesicle breakdown. This is what is called. So a microscopist, when he's examining the cell, and when he finds that there is a germinal vesicle breakdown, it begins to realize that now the cell division has started, and it is going to be dikinesis, and then of course metaphase, anaphase, and so on. Finally, the primary oocyte will divide into two secondary oocytes. Now, these two secondary oocytes are not of equal size. One of them is considerably large and the other one is very small. The very small one doesn't survive. It's called first polar body. The secondary oocyte divides again, giving rise to ootids, and one of them is very small, called second polar body. The second one survives. The one with considerable amount of yolk survives, and that finally becomes differentiated into ovum. So vitellogenesis is a vital phenomenon occurring in primary oocytes of large number of animals.